Dr. Jason Dombroski is the manager for the Cornell University Insect Collection and also for the Cornell Insect Diagnostic Lab. And today we have asked him to answer the question, I found a bug in my home and now what do I do? So we're giving him our full time today, the main presentation and the IPM minute, and we're really excited to hear his talk. Go ahead, Jason.
Hi, Amara, Matt, and everyone else. Thanks so much for the invite. I love talking about identifying insects. But I'm going to answer this question right off the bat here in a few seconds. You found a bug at home. Now what? Answer is relax. It's all good. You have to figure out what it is to see if you do have to worry about it afterwards. So what we're going to, let's see if we can get going through here. There we go. So what we're going to talk about is briefly the importance of a good ID. It should be obvious to most people why it's important to know what you're dealing with. Then we're going to cover some of the characters of what to look for when you're trying to identify an insect. And then lastly, we'll focus mostly on different identification resources that are available to you both online and with using various services. So let's start with this picture. So here's a picture of 5 different moths. Okay, let's say any of these showed up in your house. Should you panic? Well, it depends on which moth it is. If it's the one in the top left over here, I wouldn't worry. This is a very common native thing. Feeds occasionally on blackberries and roses and things like that, but it's never common enough to cause any problems. Doesn't damage any clothing or anything like that. One shows up in your house, that's just accidental, no big deal. And that hopefully is most of the insects that you encounter indoors. Because if you can get into your house, an insect surely can. There will be insects in your house. Now, well, what if it was the one on the bottom left? Well, that's a different story. This is more of a the sky is falling sort of thing in that this is a European species that is a pest of apple trees. And if it got established in Eastern North America, could be a huge problem, and that's where we would want to get some serious authorities involved to try to deal with this. But you're unlikely to encounter something like that. It's possible, but very unlikely. Now on the right, there's 3 more. These are all more typical things you would get indoors that have different ways of dealing with. So the one in the top middle, that's a clothes moth. And if you've got a bunch of these flying around, well, they're probably infesting something. It could be a rug, it could be clothing, it could be upholstery, it could even be a toy. But something that you've got to go and try to figure out the source of where these are and quarantine it or get rid of whatever they're feeding on. What if it's the one on the right? Well, those are Indian meal moths, and Indian meal moths like to feed on grains. So they could be infesting anything from crackers to rice to wheat to bird seed to dog food. They could be in almost any sort of dry grain type food. So you'd have to figure out where they're coming from. And the bottom middle one, well, if you saw that, they tend to not damage anything, but this is a moth where their caterpillars feed on dry meat. And unless you're making beef jerky in your house, you shouldn't be seeing this moth. But if you did see it, it could mean you've got a dead squirrel or rat in your wall. And so you might want to investigate and figure out what's going on there. So what I want to display with these 5 different moths here is that you really need to know what you're dealing with as to whether you panic or not, and as to where you look for potential problems. So how do you ID insects? Well, each different group of insect has different things you have to look at, but in general, what you want to do is get multiple views. So here, I just took my cell phone camera and I photographed a fly. This is just slightly bigger than a typical fruit fly. And so just to show you what I would typically do if I'm getting photos. So on the top, I've got a nice dorsal view looking down at it. And I've got another view, a lateral view, but it would have been better to be a little lower, but you can see a lot of the characters on the side. And for identifying this particular fly, I'm looking at structures of the wings veins, so I can see that in the dorsal view very clearly. I'm also looking at how the bristles are arranged on the thorax. Again, I can see that here, and structures of the antenna, which you can see here in a nice lateral view right here. So you want to try to get multiple views. Sometimes a ventral view is useful, a frontal view. So try to get multiple photos of the insect. Get clear photos / drawings. Now, these aren't the clearest photos, but they're clear enough to tell me it's a loxiniid fly and nothing I have to really worry about. Or if you're a skilled artist, Don't use artistic license. Make sure you draw it accurately, but draw the characters that you see. Another thing you want to look at is behavior, because sometimes the way an insect is sitting or acting can be very useful in separating it from others. And I'll give you some examples of that. But ultimately, very clearly capture the wing pattern venation if they've got wings, because that can be very useful. So here's some examples. Again, I just took my cell phone, took some pictures of various insects. The top left is a leaf beetle, but you can see, I can see the whole front wing cover. I can see the pronotum, the head, the antennae, the legs, including the foot structures I need to see to identify it as a leaf beetle. So it's all there in just this one shot, but multiple shots definitely would be better. In this moth, this is a moth called Papipima. I love saying that word. But with this moth, it rests with its wings very roof-like. And if you got just a straight shot from above, you'd get all the wings, but it would be distorted because you'd be sort of looking at it in an oblique angle. This way, I've shot it right, so I hit the wing sort of flat on and get a nice clear view of the wing. It also shows me the head, which is often what you need to see for moth identification, and interesting scale tufts on the body. But there's limitations because a lot of insects are very small. So here's a tiny micro wasp. This is about 1 millimeter in length, and my phone did not do a good job. I can tell it's a chalcidoid, but that's about it. I'm not going to be identifying that to species. So for something like this, you either need a way better camera, or you need to collect a specimen. And I mentioned behavior. Behavior can be a very quick and easy way to identify things. Like, for instance, how do you tell a millipede from a centipede? Well, you could count the number of legs per segment, or millipedes walk and centipedes run. It's straightforward. Centipedes are fast. You don't easily catch those, whereas millipedes are very easy to catch. So behavior can be very useful for identification. How an insect is resting. So for some small moths, some of them rest with their butt on the ground and their head in the air, while others rest with their head on the ground and their butt in the air. And that can be very useful in separating very similar species. So let's look at resources. We talked about what you want to look for. Let's start with some of the things that you can do yourself. We'll start with iNaturalist. So some of you will be familiar with iNaturalist. It's a great tool you can get as a phone app, or desktop app. And basically what it does is you take a photo of an animal, a plant, fungus, whatever, and it's a way to upload it and try to identify it and share that information with people around the world. So what I've done here, so this is a screen cap from my phone. I used the app, opened it up, and I took a picture of this moth. And you'll see it grab the date, the location, all that's there. And then if I click on what did you see, the AI then figures out what it sees. And this photo is from 4 years ago, but the AI has gotten so much better even in the past year that I'm certain it would identify this one confidently to species. In fact, most of the insects, if I can get a clear photo of it that I encounter on a regular basis, the AI on this is good enough to identify it, at least pretty close to it. So really useful tool. Plus, as you're uploading these onto the web, you'll get experts that will go and verify your identification, or change it if it's wrong. And also you're contributing to science. So from my end as a scientist, I'm using data uploaded by people all the time on iNaturalist. Often when they found just some random bug indoors, it can be something really significant. You can also use iNaturalist sort of on your computer. Let's say, for instance, you went outside this afternoon. It's a beautiful day here in Ithaca. You went outside and you saw a little blue butterfly, and you couldn't catch it. You got kind of a decent look at it. But what is it? It's a blue butterfly. Well, you can go to iNaturalist, and what I did is I selected Tompkins County, I selected butterflies, and in the filters, I selected October. There's not a lot of butterflies out now. Here are the butterfly records for October for Tompkins County. It wasn't a monarch. It wasn't a small white or clouded sulfur. Oh, eastern-tailed blue. Okay, well, it could be that. And so then you can click on eastern-tailed blue, takes you to the page for it. There's the time of year you can find it. Yeah, it's still out in October, not very commonly. And the upper side is bluish. Okay, that's good. And you can click down and see, okay, it's definitely found in New York State, all over the place here. And there's further information. So you can do identifications like that. And admittedly, I do this regularly when I know it's going to be one of these several species. Okay, select the time of year, the location, that can help narrow it down for you. So the point I want to get across here is iNaturalist is an amazing tool for identification. Let's move over to bug guide. So for those of you that have done insect identifications longer, you'll be more familiar with bug guide. This site currently is used less by naturalists, but it's a great resource for more in-depth information. So let's say you figured out, okay, it's one of these several species, and let's say you have a grasshopper, okay, and you figure out it's the genus Melanoplus, one of the spur-throated grasshoppers. Okay, they look kind of similar. How do I separate them? Well, you go to bug guide, you go to the genus you want, and then you can scroll down. There's some pictures, and then there's all this information. So there's so much information here put on by experts on how to identify things, how difficult things are to identify, how easy they are, etc., and a lot of additional information. So bug guide is rich with stuff like that. You can also go through and browse. So if you want to browse through the spur-throated grasshoppers, here are the different species. You can even narrow it down by geography. So if you want to just look at the New York species, you could do that. And then, of course, any of these you could click on and find further information. So it's really quite a useful tool. You can also contribute to Bug Guide. You can upload photos and have experts vet it. But again, it's used much less frequently than iNaturalist currently. Okay, so let's say you've tried iNaturalist, you weren't happy with the answer. Bug guide, yeah, you couldn't quite figure out what it is. It seems like this might be difficult to identify. Well, you got to get a specimen then. And so first things first, be careful not to damage the specimen. From my perspective as someone that identifies insects, simple thing like breaking hairs off of a caterpillar makes it much more difficult to identify things. Moths and butterflies are covered in scales. The more they get bashed around, the more those scales come off, and the harder it is for us to identify them. And very importantly, put in a crush-proof container. We get pretty regularly people that find an insect in their house. They want to submit it to us for identification. They put it in an envelope and send it through the mail. And when they put it in there, there was a beetle. And when we get 1,000 pieces of tiny insect. So don't just put it into an envelope. It's got to be in a crush-proof container. I'll give more on that later, because no matter how many times you write fragile on an envelope, it's going to get crushed. And then as far as you generally don't want to send live stuff, especially if you think it's a bed bug or something. We don't appreciate getting live bed bugs or lice and things like that sent to us. You can kill it in the freezer. If you want to just get photos of it, you can chill it in the fridge. Fridge is a great way to slow down the insects so you can get some nice photos. But yeah, killing them in the freezer works quite well. Okay, so first place you could try is your local cooperative extension office. So basically, you could come in there with your specimen, and quite often the people there will be able to recognize a lot of the common stuff and be able to render advice if necessary as to what to do about the insect. If they aren't able to help, they might recommend us in the diagnostic lab, and you can also go straight to us. The insect diagnostic lab, we do charge $25 a sample for identification, but we do have rapid turnaround for routine things that we see a lot. Basically, usually when we get it, we can identify it within a day or two sometimes. So typically it's rapid turnaround. Some things are more difficult to identify, so it might take us longer. Things like mites and aphids and thrips will take longer. Scale insects take longer, but usually we get pretty rapid turnaround. And then what we'll do is we'll get you the identification and often send you fact sheets. Actually, if you even browse on the Insect Diagnostic Lab webpage, you'll see we've got a whole list of fact sheets that you can check out that will give you advice on just general things like how to deal with beetles infesting woolens and things like that. But we have a lot of resources there and a lot of links there. So yeah, so we'll get you an AD. We'll send you appropriate fact sheets or information if there is no fact sheet, and it's all good. And as far as numbers, we're a pretty busy lab. Last year was a slightly slower year. We had 490 public submissions, but many of those had multiple identifications. We also have contracts with New York State Department of Agriculture and Markets, where they have us do identifications. We did over 8,000 for that. And then we're also the identifiers for European cherry fruit fly, which is a recent invasive here. And we identified 6,000, almost 7,000 traps of those. And that involves looking at hundreds of flies quite often on these traps. So that's just an incalculable number of IDs. But luckily, we've got a great team of technicians and students here that can do a lot of this work quite efficiently. Okay, so this is straight from Kevin, who's a co-author on this talk, Kevin Skimmerhorn, who basically manages the lab. And if you contact the Insect Diagnostic Lab, you would be directly talking to him. Basically, here are sort of some of the guidelines he set out for a submission. And that is, number one, the most important step, send us an email. Start with that. Send us an email, put some photos in, say, hey, I've got this. You know, I'm thinking of sending it as a sample. And from there, sometimes we can identify it. And on our website, there's a form you can fill out. And you know, sometimes we'll be, oh yeah, I don't need to send that. We know what that is. Here's what it is, and you're all good. Sometimes we'll say, okay, yeah, no, you'll have to send that in. That's a tougher identification. So yeah, take photos and email. And remember, we've talked about photos and how important it is to get a nice crisp photo. I'll show you some examples. Package them properly. We talked about that, and then ship it to us. So for the email, yeah, I basically said it helps determine if you actually need to send it in, because sometimes we can't ID it. Also, it lets us know to expect a package so that we can be ready for it when it comes. And there's the email address right there, IDL Diagnostic Lab at Cornell, but just go to Insect Diagnostic Lab, Google that, and you'll find all our contact information there. Going back to photos, so here's some examples he put up. So good photos on the left. The only one I'd complain about in the left one is I would like a dorsal view as well. That's the ventral view, the underside. So it's good to get multiple views, but these are nice crisp photos where we can pretty clearly tell what things are. These are not on the right. So basically blurry photos, if it's that small, just send it in. But you can try anyways. Okay, collecting and preserving. This is important stuff. So if you've got a hard-bodied insect, so think of, I hate to put it this way, but think of something if you step on it, it goes crunch. So it's hard-bodied. Collect it into a container. Ideally not a Ziploc bag because those get crushed. But basically make sure, again, it's in a crush-proof container. And then freeze it so you don't send us live stuff. And then what you can do is wrap the specimen in paper towel or tissue. That makes sure it doesn't bounce around because that just causes them to break as well. Excuse me. And if there's broken parts, include that as well. For soft bite insects, it's slightly different. If you weren't sending it through the mail, we'd ask it to be an alcohol. Isopropanol, that rubbing alcohol works fine. Ethanol is better, but not everybody has access to that. And basically what you can do, put it into a tightly sealing container. And then you'll want to put a cotton ball or a piece of tissue that's soaked in alcohol. The reason why you don't want to just dump alcohol in there is that you can't send that through the mail legally because it is flammable. But a cotton ball or tissue soaked, that's just the right amount. That's perfectly legal. But you do want to make sure you take that vial up very carefully, that container up. And again, that's crush proof. And that you then put that into a Ziploc bag so that it's sealed up really well and it's not going to leak out. And then send it off. So proper packaging, more on that. Never send just in a flat envelope. Again, I told you that we end up with many, many pieces, and it's just a real pain to try to identify something. And usually we end up saying, just try again, send another one, but in a crush-proof container. Smaller boxes are better. If it's a small box or a small container in a big box, put some newspaper or bubble wrap or something so it doesn't just keep bouncing around and it doesn't get, the box doesn't get damaged in shipping. Padded envelopes you can do, but again, it's got to be in a crush-proof container. And shipping, our address is available on our website where you can just mail it to us. But again, an email first is good to let us know that we're to expect it. And there is a form that you've got to fill out because we do need information on where it's from because I don't know if someone's sending something from New York State or from Patagonia. And that's very important in identification, knowing where the specimen came from. And then, of course, include your payment in that as well. There's full information on that on the website. And with that, that's all I've got to say. So I'm happy to take questions.
Thank you, Jason. That was great. I think the first question that I wanted to look at And folks can continue to keep putting questions in the chat. Someone asked, is iNaturalist the same as the Seek app? They have had some misidentifications with the Seek app when they've used it to try to identify things. What are your thoughts about that?
Yeah, so I don't know the full relationship. Seek is, I think, within iNaturalist. So it's using the same algorithms. And yeah, it does make errors. What I will say is it makes fewer errors than Google Lens, because when you look at it, or any sort of online search where you're searching for images, iNaturalist's AI is looking at just specimens that are research grade, and most of those have been vetted by experts. So you've got expert identified things. Google, when it's searching an image, finds everything online that looks like that, and then whatever name people have called it. And a lot of them are Picasa images, so people have uploaded their albums and called things whatever they wanted, and those errors get perpetuated. So while Google will often get it right, iNaturalist gets it right more often, but it still does make mistakes. You can't completely depend on it. It's always good to verify, like, just to make sure, does this make sense? I also find that if you want to increase or decrease the error rate on iNaturalist, just get a really clear picture where it's filling most of the screen. If it's a tiny little black speck, no matter what it is, it's going to call it a light brown apple moth. Um, if it's this big, it will not call it a light brown apple moth unless it is a light brown apple moth. So getting a clear photo is very, very key.
Looks like someone is curious about that picture on your thank you slide. They want to know if that insect is a bee.
It is not a bee. It is a mimic, though. That is a bee fly, one that's called villa. And so bees have that black and yellow patterning to make them so that animals know that they could sting and can harm them. There are so many different insects that mimic that are actually harmless. There's a lot of beetles that do it, a lot of flies that do it, even some moths that mimic bees, really convincingly so. In fact, I'm really quite embarrassed. I was leading a moth walk once, and this little princess wasp flew by, and I even knew which genus it was. And I said that, and one of the participants said, "You sure that's not a moth?" I'm like, come on, I study moths. I know that's a wasp, and I caught it, and that was a moth. I was completely fooled by it. So they can be very impressive mimics. That particular bee fly you can find pretty commonly across New York State. Midsummer, they visit various flowers. They're also pollinating just like bees do. In fact, for some flowers, flies might be more important pollinators than bees.
Very cool. There's a question wanting to clarify the costs for the insect diagnostic lab if you are a master gardener volunteer through Cornell Cooperative Extension, and I think we have a lot of master gardener volunteers here. So if a master gardener volunteer sends an email to the lab saying, we got this sample into the Cooperative Extension office, how do fees work in that situation?
Basically the same way. So we do get a lot of referrals from master gardeners. And so you would basically have the submitter fill out the paperwork, or you can do it too. I think we do have a separate form for master gardeners, for CCE people to fill out. But basically someone's got to pay. We need to feed our families somehow. But yeah, so it's still $25. And I should clarify, um, it's $25 whether you send us in one fly or 20 flies, as long as you think they're the same thing. And it's often good to send duplication in, because quite often one will break, or sometimes you can only identify males and you can't identify females, or vice versa. So it's good to send in multiple. We're not going to charge you 20 times that. Now, if you think you've got 20 different things, well, then that's a different story. Then we'll have to talk. And we might be able to get you a discount on that rate. But yeah, send in multiple samples. That's key.
And someone is asking for a quick reminder of the other apps or websites besides iNaturalist that you were suggesting.
Bug Guide is a big one. So that's the one I briefly mentioned. It's just chock full of information for insects of North America. BUGGUIDE. So that's got a lot. And then beyond that, there are specific websites for specific groups. So for things like moths, there is the Moth Photographers Group, which is an excellent resource and has photos of almost all the moths of North America. If you're looking at, butterflies, there are various butterfly websites. Like the Butterflies of the Americas, which has lots of information from both North and South America for butterflies. Say it all depends on the group. And if you want to do a deeper dive, sometimes you'll find more specific papers. The good thing with Bug Guide is it'll often link to these other more advanced resources. So for instance, let's say if you want to look at hoverflies, um, you'll go to Bug Guide, you can do what you can there, and then it'll point you to websites, but also books. There's a recent a field guide to the hoverflies of Eastern North America. If you wanted to do bee flies, like on the last slide that I had, there is a lovely key for the bee flies of Ontario, which covers mostly New York species. So all these references are listed on Bug Guide.